Welcome everyone, we're so happy to have you here at Smithsonian Science How. We're kicking off the first show of the brand new season with invertebrate zoologist from the Smithsonian's National Museum of Natural History, Dr. Dave Pawson. Dave, thank you so much for joining us here today.
Thanks for having me.
So Dave, you're an invertebrate zoologist and you brought along some of the collections that you study, like sea stars, I see sea urchins. These are just fabulous. Can you tell us what they're called?
Well, as a group, they're called echinoderms, which comes from two Greek words meaning spiny skins. And most of them, as you can see, have spiny skins indeed.
And we can see some of them on the screen here. They come in all different shapes and sizes and colors.
Yes. Yeah, there's a great variety of them. There are about 7,000 different kinds alive today in the world's oceans.
And I wonder what beyond having spiny skin unites this entire group. There's clearly a lot of diversity here.
Most echinoderms have hard skeletons, bony skeletons, hence their name. But one group of echinoderms called sea cucumbers have squishy bodies. And they have squishy bodies because they do have skeletons, but the skeletons are reduced to little tiny microscopic bones in the skin. So it's hard to believe that this animal is related to a starfish, but it is.
So what are the other characteristics? What are some of the other traits that unite echinoderms?
One of the obvious ones is that they have usually have 5 parts to their bodies, 5 so-called arms. Even a sea cucumber doesn't often looks as though it doesn't have 5 parts to its body, but if you look inside, you'll find 5 of various things. And it's not so obvious in some of these animals, but it's certainly obvious in the starfish and in these so-called brittle stars, which have 5 arms coming from the mouth. In the sea cucumbers, the little bones in the skin, as I mentioned, are microscopic. They're very beautiful. They're fun to watch. These animals are fun to study under the microscope, at least. Another characteristic of echinoderms is that they can grow new parts. When a part of their body goes missing, if you cut a starfish in half, it will grow into 2 starfish. And some of these animals can lose body parts very easily. And another characteristic of echinoderms is that they have so-called tube feet, which are hollow organs filled with fluid. And at the end of the tube feet is a disc-like termination there. And they use these feet to help them in getting around, locomotion, and feeding, and they're also used for breathing too. Some tube feet are used as lungs.
And here, what's happening in this picture?
Oh, that last picture, a starfish is feeding on a coral in the deep sea.
Interesting. So echinoderms have been referred to sometimes as stars of the deep sea. Why is that?
Mainly because there are so many of them down there. Echinoderms live all over the world, in all the world's oceans, from the seashore down to the greatest depths. And when you get into deep sea, they really come into their own. In fact, on the deep sea floor, where it's dark, totally dark, sea cucumbers can make up more than 90% of the total weight of animals on the seafloor, deep sea floor.
And do they have a specific role on the seafloor?
Sea cucumbers do. They feed on mud, most of them. They're like earthworms. They pass mud through their bodies, and as it passes through, they add oxygen to the mud, and they make the mud more livable for lots of other kinds of animals that can then settle in, like worms and little shrimps and so forth.
A critical role indeed.
Yes.
What's so fascinating is that not that long ago, over 100 years ago, scientists didn't even think anything lived in the deep sea. Isn't that right?
Yeah. Up until the early 1860s, most scientists believed that the deep sea was essentially a desert below a depth of about 1,800 feet. But in 1868, a Norwegian scientist called Dr. Michael Sars, took a dredge haul in a very deep part of a fjord, inlet in Norway, and he pulled up this strange animal which is called a sea lily. And he made two famous discoveries in collecting this little animal. One was that he was able to prove that these sea lilies are still living in today's oceans. They were only known as fossils until that time.
So we can actually credit echinoderms for helping us discover life in the deep sea.
In the deep sea, yeah. And he also proved that there was life in the deep sea. And a lot of scientists became very excited about these discoveries because they speculated that maybe the deep sea was a place where a lot of living fossils were hiding. Later work has proven that this isn't so, but there are still lots of weird animals living down there.
This is very incredible. It does look a little weird as well to think of that as an
yeah,
animal.
Yeah.
So tell me, the discovery of these sea lilies in the deep sea, did that kick off a frenzy of deep sea exploration?
Yes, it certainly did. Many countries, especially in Europe and also the US, decided that exploring the deep sea was a good thing to do. And many very large purpose-built research ships were constructed. Among them a very famous American ship called the Albatross, which was launched in 1883. She was a steamer, but she also could sail when she needed to. And she worked for almost 40 years in the Atlantic and Pacific Oceans and sailed about 1 million miles and collected tons and tons and tons of animals.
Which are now here in the Smithsonian in our invertebrate zoology collection, correct?
Yeah, we have enormous numbers of them in our collections.
There was one scientist in particular that looked at echinoderms and collected a lot of them aboard the Albatross. Who was that?
He was a man called Austin Hobart Clark, and he was invited as a naturalist to participate in the 1906 cruise of the Albatross, which went from San Francisco to Alaska and around Japan and back to San Francisco. The entire trip took 7 months. And Austin had just been married to his wife Mary for 1 month before he took off on this long trip.
It's a very kind wife.
Yeah, their families were not too happy about this, deserting his spouse after 1 month, but they decided it was probably a good career move to take this job for 7 months. And so he did it. While he was on the ship, he wrote long and very affectionate letters to his wife, Mary. And there were hundreds of these letters.
Wow.
He wrote them pretty much every day.
A whole folder of them.
We have great folders full of them. And my wife, Doris, has transcribed them now, and they're about to be published. And they're not just affectionate love letters.
I can see it at the top. Dearest, dearest, dearest sweetheart.
Yeah. They are great diaries about life on board the ship, day-to-day life, describing all kinds of events that happened, and also describing the ways in which they collected animals, what the animals looked like when they came up in the nets and so forth. It's a wonderful, detailed diary. And we're very glad to have this record of life on board a ship 110 years ago.
So this record of life aboard a ship, as you say, is providing you more information about the animals that were collected and life on board the Albatross at that time.
Yeah, that's right.
Can you tell us a little bit about what life on board that Albatross was like?
Well, it was, in many ways, it was a rough life. It was a very lively ship in rough weather. And Clark told a bunch of stories about how they collected animals in all parts of the ocean, from the surface, such as shown here, down to very great depths. And they would tow these enormous nets around the bottom in great depths and pull up literally tons of animals, which would be sorted and carefully preserved.
Now, in addition to the scientific value of these love letters, essentially, that you've been able to find, is there any kind of anecdotal information that's within these letters that provides what life was like, maybe the daily, the relationships aboard the people, among the people on the ship? Yeah,
that's, some of the stories are just amazing. One of the very exciting aspects that Clark dealt with a lot was a mascot on board the ship called Buck. Buck was a very large goat. And in those days, ships had tended to have mascots, dogs and cats and monkeys and so forth.
It's a big goat.
But Buck was very large, and he was on the ship for at least 12 years that we know of. And he was sort of a nasty animal in the sense that if you were to tease him, he would get you later. He was vindictive. And there was a story about a cabin boy carrying a very large tray full of meals from one part of the ship to another. And apparently he had teased Buck earlier that day or earlier that week. And Buck saw his opportunity and came and hit this young man from behind. And he went sprawling and the meals went flying everywhere.
Oh dear.
Yes.
So in addition to providing some humor about what life was like aboard the Albatross, Austin really provided context for the objects that are here at the Smithsonian now that are still being studied. But
Yeah.
what did Austin go on to do after he got off the Albatross?
Well, he started his life as a collector of birds. He was a fanatical collector of birds. But as he became more and more involved with deep sea trawling, he became interested in these particular animals, the echinoderms. And he, one of his early, I think he was about 12 years old when he did that.
A great illustrator.
Painting. Yeah, and so when he came back after the cruise to Washington, he studied echinoderms for a year, working for the National Fisheries Service. And then he was hired here at the museum in 1908 and became a scientist. And he was here for the rest of his life until he retired in 1950.
And here you are, side by side with Austin.
Yes, I feel as old as he does right now. But yeah, we're holding exactly the same animal there. It's extraordinary.
So he left behind not only his letters to provide context with what happened during this very specific research trip, but he left behind his research, his science, and a lot of collections for you to study as the echinoderm curator here at the Smithsonian.
Yes, that's right. And he was a great scientist. He described about 500 new species of echinoderms and published hundreds and hundreds of scientific papers and hundreds of popular magazine articles as well.
How wonderful.
He was a great communicator.
Thank you for introducing us to these echinoderms. Let's get to some of our student questions.
Okay.
This first one comes from Wallenpaupack Area School District. Thanks for coming, an alma mater of mine, actually.
Aha.
So how many feet does a starfish have?
How many feet? Well, some of them have thousands of feet. The feet, if I can lift this up for a moment.
Absolutely.
This animal, for example, has the remains of the feet are dried here. He has hundreds of feet underneath each of these arms. And he uses them for holding on to very hard areas on rocky shores, rocky coasts, and also for feeding on mussels and clams, using the feet to pull apart the shells of mussels and clams.
Great question. So this one's from Gabe.
Yeah.
Are there any sea stars that are endangered?
Yes, and it happens that some of these sea stars, including this particularly large type, is endangered in California and to the north of California because it's suffering from some disease which is called a wasting disease. And it's pretty much has died out. The disease has died out pretty much by now. But for a few years, literally thousands of these starfish were being killed by this strange malady.
It's unfortunate. So our next question comes in by video, so let's take a look at the screen.
Hi, I'm Sydney. Hi, I'm Diamond. Why do they call you sand dollars? Yeah, why do they call you sand dollars?
I think they're interesting. Those sand dollars are either from New England, somewhere up north, or somewhere off Washington State or Oregon State. I think they were called sand dollars because small ones would wash ashore, and they looked probably a little like a $1 coin when they washed up. And there are a couple of very large sand dollars from down off of the coast of Florida.
Wonderful. So students, keep your questions coming. We'll get to more of them but first we're going to learn a little bit more about the history of deep sea research and Dave, your work here at the Smithsonian. So tell us, when Austin and the other scientists were collecting specimens from the deep sea using big nets, Were there any major limitations to that methodology?
Yes, I think the largest limitation was the fact that a lot of deep sea animals are very fragile because they have a lot of water in their bodies. And when they were collected in nets along with rocks and all kinds of hard things, they'd become mangled as the net would come up to the surface. And so when these animals were dropped onto the deck of a ship and then put into bottles and preserved, it was often difficult to determine what they looked like when they were alive. And Austen, in some of his letters, for example, tried to reconstruct what these animals might have looked like. And he was pretty good at it, but there were a lot of things that we didn't know until we were able to do more in the way of sampling these animals.
What came after trawling these huge nets along the ocean floor?
Well, the next big development was the introduction of cameras into the deep sea, dropping a camera on a line from a mothership to the seafloor.
Which we see here.
And when the camera would hit the seafloor with the motion of the ship on the surface, an electronic flash would go off and the camera would take a picture. And over the course of 2 hours, the camera could take 800 pictures. And millions of these photographs were taken. And at last, we were able to see how these animals are making their living, what they're doing, how many of them there were. We tried to interpret their behavior, often completely incorrectly. And millions of these pictures were taken. And I probably looked at more than 1 million of them over the years.
Wow.
Very exciting.
It's a first look at the deep sea.
Yeah.
So what came after this camera apparatus?
The next development was especially during the 1960s and beyond, and that was manned little submarines, submersible so-called, which could take between 1 and 5 people down to great depths. And here is the chance to go down to where the animals are and see them doing their thing and videotape them and photograph them and pick them up if you needed to study them more in the laboratory. This shows my wife on the left and Dr. Carol Baldwin on the right, about to descend in a submersible in Curacao.
Now, Dave, how many subdives have you done?
I think I've made a few more than 200 dives over the past many years.
That is amazing.
40 years.
Now,
Yeah,
that means that you've seen
very lucky.
parts of the ocean that nobody else has seen before. What does that feel like?
Oh, it's always exciting. There's always something new to see when the submersible gets to the bottom and the pilot turns the lights on. Suddenly things are illuminated and there's always something new to see.
How fabulous.
It's incredibly exciting.
Now, after the submersibles, there was another development in deep sea. What was it?
Well, it's going down a submersible can be dangerous. It's not as dangerous as riding in an automobile, but the trend has been over the past several years to send submarines down that don't have people in them, and these are called remotely operated vehicles. And an ROV, you can sit on board the mothership or you can sit on land and watch what the ROV is finding. And the ROV can videotape animals, it can photograph them, and it can also collect them.
Just like this animal here. What are we seeing?
Yes, I wish we'd collected this one. This was a photo taken down in the Gulf of Mexico. It's a sea cucumber, which has decorated itself with dozens of little triangular-shaped shells from a little mollusk, which is about 1 inch long, related to clams and oysters and snails. And this little animal swims vertically in the ocean, swims upwards all the time. And they're all over the world. When they die, they drop to the bottom, drop to the sea floor, and the soft parts rot away, leaving the beautiful, clean white shell. And the sea cucumber picks these shells up and attaches them to its body, holding on with its tube feet.
How fascinating that it can sift and find those out of everything down there.
We have no idea why they're doing it. The decoration, it's totally dark down there. The sort of white shells, if there's any light down there, would tend to reveal the body of the animal rather than conceal it.
So I'm sure you have a lot of other hypotheses as to why they would do this, because as a scientist, I think you have discovered why some shallow water species that are related, so sea urchins, their cousins, sea cucumbers' cousins, they also cover themselves in tide pools, and you do know why that happens, correct?
Yes, they do. Other scientists have discovered why these animals cover themselves, and mainly the sea urchins in shallow water cover themselves with debris to reduce the ultraviolet light from sunlight that's hitting their bodies, and it could damage their bodies.
So you told us that it may not be for camouflage because they live on the bottom of the sea in really deep water.
Yeah, the deep water ones. Another thing the shallow ones do that's fairly common is they hoard food, drifting pieces of seaweed. They'll grab them and hold on to them and then pass them down to their mouths when they feel like a snack. We were amazed to find that in the deep sea, in the Bahamas, southeast of Florida, there are 33 species of sea urchins there that we were studying, and 3 of them cover themselves. They pile debris on tops of their bodies. And we don't know why. There's no sunlight down there. There's no ultraviolet radiation. The debris that they're using to cover themselves is not material that they would use as food. It's a mystery, and it's something we haven't been able to. We've brought live animals back from the deep and tried to get them to cover themselves, but they won't cooperate. So we have no idea. We think that the animal, possibly the animal himself, doesn't know why he's doing it, or she. It may be that the covering activity is genetically linked to something more important that the animal must do. We don't know what that might be.
So this is still a mystery out there.
Still a mystery. There are a lot of mysteries in the deep sea, for sure.
So Dave, you're a modern scientist, but we have a lot of historic collections here on the table. Is there any scientific value in the objects that came to the museum 100 years ago from expeditions like the USS Albatross?
Yeah, these collections that are quite old are still very scientifically valuable, and scientists come from all over the world to study them. In this particular case of that sea urchin there in front of the table, this sort of pancake-like object, we looked inside some of those and found that their intestines, their stomachs and intestines were full of pieces of what's called turtle grass. And here's the same animal alive in a depth of about half a mile down off of the coast of Florida. And we were amazed to discover that this animal, this particular species, which can be present in very large numbers, feeds on turtle grass and essentially nothing else. So turtle grass is its preferred food. And turtle grass grows only in very shallow water around the coast of Florida and the Gulf of Mexico. And it's like a coarse lawn grass, and it can be found in water that ranges from 1 foot to about 30 feet deep, very shallow. And with wave action and storms, pieces of turtle grass are broken off, and they float out to sea, and they become waterlogged, and they eventually sink down to the deep sea floor, where these urchins are waiting to feed on them, and also other kinds of animals too, because food is always scarce in the deep sea. The problem for these urchins is that the turtle grass beds are disappearing, mainly because of human activity, sort of development, building of marinas and so forth, but mainly fertilizers and other chemicals that are brought down in rivers and streams into the shallow depths, killing off turtle grass beds. And we've learned that over the past 40 years or so, the beds have decreased in size by about 70%.
That's devastating.
Which means that the food available for these guys that are living in very deep water, these urchins, has decreased by 70% also. Eventually they're going to have to change over to another diet. We don't know what that might be. But I've always been struck by this connection, because here is something that we are affecting in water that is 1 foot deep and right there where you're wading. And what's happening out there a mile out to sea and a mile down you're directly affecting these sea urchins by killing off a turtle. It's this direct connection that's always impressed me greatly.
Absolutely. How closely connected we actually are on land in the shallow sea to these deep water specimens.
We are indeed.
So we have a lot of questions coming in, but before we get to them, Dave, can you tell us about some of the recent discoveries you've made? You've shared a couple mysteries out there for now.
We have, here is an example of a sea lily related to that one that Dr. Sars found in 1868.
How beautiful.
It's a dried one. We've discovered a new one, and it's not nearly as pretty when it's dead. There it is. But it's very beautiful when it's alive. And I hope we'll show a picture of it soon. But there it is there. Gorgeous animal. It's only about this high when it's fully grown. And we described, a Russian colleague and I described that. And here's a polka-dotted little worm-like sea cucumber that we described from some methane seeps, so-called. It's holes in the seafloor where methane gas is coming up in the Gulf of Mexico. Here's another kind of sea lily that's about 2 inches high that we described some years ago. And our latest little find is this guy, which is a brittle star. It looks rather like a starfish, but it's a brittle star. It's a female. It has 10 masses of eggs there that you can see. It's about ½ an inch across. And it was first discovered off Curacao. This reddish animal was attached to a bright green beer bottle lying on the seafloor.
It wanted to be discovered.
Yeah, it was.
Dave, thank you so much for being here and sharing all of this wonderful information about sea stars and urchins and all their relatives.
Well, thanks. It's been an honor to be here.
Where can our viewers learn a little bit more about echinoderms?
Well, I'd recommend the first place they go to is a blog run by one of our resident scientists, Dr. Chris Marr, and it's called the Echinoblog, ECHINOBLOG, and that has some wonderful information about echinoderms in it.
Wonderful. Thank you so much, Dave. And if you want to learn more about the history of deep sea research and the ocean in general, you can go to our Smithsonian Ocean Portal. And we also have teaching resources related to Dave's work on our Curious website at curious.si.edu, which is the same place where this program will be archived later this evening. Thanks again for joining us today on Smithsonian Science How. We hope to see you next time.